Hi everyone, my name is Florencio Gobellan and I'm so excited to share my research on rest and state brain connectivity and the role of depression on pediatric epilepsy using fMRI. So, depression is a commonly observed psychiatric condition in epilepsy, especially including in children and adolescents. While epilepsy can result in both structural and functional brain abnormalities, depression is primarily linked to functional changes in brain networks. A way that we can study brain networks is through something called the triple network model, which includes three major networks, which is a default mode network, which is associated with self-referential thoughts, the salience network, which helps process important information and emotions, and lastly, the central executive network, which involves attention and decision making. Previous research shows that there is an issue with connectivity um, in those depression when we talk, I'm sorry, that there is um, issues in connectivity in these networks when depression is present, but we wanted to explore how epilepsy and depression interact at the functional connectivity level, specifically in adolescents. We studied 37 adolescents between the ages of 10 to 19 years old. 13 of them had epilepsy, 24 were typically developing controls, 16 of them were females. Each participant underwent a 10-minute resting state fMRI scan while keeping their eyes open. Um, to measure depression severity, we use the 9-item patient health questionnaire, which is PHQ-9 for short. And we analyzed 19 key brain regions, which are all between the three models, which is the default mode network, the salience network, and the central executive network, to compare connectivity between the differences. So we are um, comparing epilepsy versus typically developing individuals and individuals with and without depression. So we had some three key results. The first one is we saw higher connectivity in epilepsy. So compared to typically developing individuals, those with epilepsy show higher connectivity between the default mode network and the salience network and the central executive network. And this probably suggests disturbances in how the brain balances internal thoughts and external attention. The second key result is depression does impact connectivity, specifically in those with epilepsy. So within the epilepsy group, those with depression have lower connectivity between the salience network and other brain networks, particularly in the prefrontal cortex connections of your brain. Compared to typically developing individuals with depression, epilepsy patients with depression show reduced connectivity between the salience network and the default network, as well as changes in the central executive networks. Basically, this is telling us that depression does have a major impact on connectivity within the epilepsy group population. And then lastly, we saw differences in brain network partners. So we typically develop in individuals with depression show reduced connectivity within the salience network, but higher default mode network to the central executive network connectivity. So between those two. Um, in contrast, epilepsy patients with depression had higher connectivity between the stimulus network and the central executive network, particularly in the prefrontal cortex, suggesting some kind of disruptions in how the brain processes important and task-related information. This findings highlights major differences in how brain network functions in epilepsy within and without depression. Understanding these network disruptions can help us in three major things. First of all, identifying biomarkers for altered brain function in epilepsy. Second, it can help us develop targeted interventions to improve mental health outcomes in kids. And lastly, it helps us track changes in brain connectivity to refine treatment for adolescents with epilepsy and depression. This research is an important step toward bringing more attention and just bringing more awareness to epilepsy and mental health. I'm incredibly grateful for my support from my mentor, Dr. Krista Cooper and Dr. Tracy Greer, and the McNair Scholars Program and Cook Children's Medical